Hey there, lab geniuses! Welcome back to the Genius Review Center, where the science sticks. Just like today's topic, phosphodiester bonds. Now let me ask you a question. What do you think keeps the DNA strands intact? Magic? Glue? Cramming the night before? Wrong! That's what we're gonna talk about. Today, we're gonna have a close-up look on the molecular handshake, nay, the chemical pinky promise that keeps the DNA strands intact. The phosphodiester bonds is what keeps the DNA and RNA from crumbling like a horrible group project. Let's set the stage. The DNA and RNA are made of nucleotides, the little building blocks of life. Each nucleotide has three components. You have a nitrogenous base. This is made of adenine, guanine, cytosine, and thymine if it's a DNA. Adenine, guanine, cytosine, and uracil if it's RNA. It's also made of a 5-carbon sugar. This is a 𝐷𝐞𝐳𝐢𝐴 if it's DNA, and it's a ribose if it's an RNA. Last but not the least, you have a phosphate group, which may contain more phosphate groups, depending, and these do the binding magic. Now, if you ever forget the difference between a nucleotide and a nucleoside, here's a genius trick. Tide has more. The nucleotide has a phosphate group while a nucleoside doesn't. Easy peasy. Now look closely at the sugar of a nucleotide. We only care about 2 carbon atoms. The 5-carbon atom, which contains the phosphate group, and the 3-carbon atom, which contains the hydroxyl or the OH group. Now this is where the magic begins. A phosphodiester bond is formed when the 3-OH group of the one nucleotide and the 5-phosphate group of the next nucleotide bind up. They link up in a condensation reaction, meaning a water molecule is released. Then boom! You now have a strong covalent bond forming the sugar-phosphate backbone of DNA and RNA. The sugar-phosphate backbone always builds in one direction, from 5 to 3. The directionality is crucial for DNA replication and RNA transcription. Enzymes read and build nucleic acid in the 5 to 3 direction only. Alright geniuses, let's throw DNA and RNA into the ring. Who's got the stronger backbone? The 2-OH group in RNA makes it more reactive and less stable. And the DNA, on the other hand, has no OH group, making it stable and reliable and built to last. Let's do a mental activity. Picture a nucleotide chain. Now grab your mental pencil and label the sugar, label the phosphate, and find the 3-OH and 5-phosphate groups. Connect them, and that's where your phosphodiester bond goes. Boom! Science! Magic right before your eyes. Alright, smarty pants, let's see what you've got. Quick 3-question quiz. Question 1. What is a phosphodiester bond? We have A. A bond between nitrogenous bases. B. A hydrogen bond between DNA strands. C. A covalent bond between the sugar of one nucleotide and the phosphate of the next. Or D. An ionic bond. I'll give you some time. Correct. It is C. A covalent bond between the sugar of one nucleotide and the phosphate of the next. Question number 2. Where exactly does it form? A. Between nitrogenous bases. B. Between 3-OH and the 5-phosphate. C. Between ribose and the base. Or D. Between DNA and your dreams. Correct. It's B, between the 3-OH and the 5-phosphate. Question number 3. Which is true about DNA versus RNA? You have A. DNA is single-stranded. You have B. RNA is more stable. You have C. They have identical sugars. Or is it D? DNA has deoxyribose and RNA has ribose. Correct. DNA has deoxyribose and RNA has ribose. If you got 3 out of 3, you're officially a genius in bonding. To recap, phosphodiester bonds are connected nucleotides via the 3-OH and 5-phosphate groups. They build the sugar phosphate backbone of both DNA and RNA. They are formed in the 5 to 3 direction and are in the same DNA and RNA, but their sugar give off a totally different vibe. So the next time someone tells you you've got strong genes, you say thanks, it's the phosphodiester bonds, baby. This has been your genius guide to getting bonded. Stay smart, stay curious, and remember, chemistry is what keeps us all together, literally. Bye-bye!